Hello there and welcome to the Dreamy Ocean Channel. As the day's last light softens and the world outside begins its quiet descent into night, allow your own body to gently follow. Release any tension held within your shoulders, letting them softly drop, and feel your jaw unclench, easing into a peaceful repose. With each slow, measured breath, imagine any lingering thoughts or worries drifting away, like distant clouds dissolving into an endless, tranquil sky. Feel the gentle weight of your limbs settling into comfort as if cradled by the very earth beneath you, inviting a profound stillness. Tonight, we journey into the cool, silent depths of our planet's vast, shimmering oceans to encounter a creature of breathtaking beauty and profound mystery. A jewel of the reef whose diminutive form holds a power that whispers of the ocean's most ancient, untamed secrets, reminding us of the delicate, potent forces that exist just beyond our everyday sight. Let us now together slip beneath the surface of the waking world and allow the gentle currents of this story to carry you softly into the quiet embrace of slumber. The ocean's silent jewel. An introduction to the blue-ringed octopus. Deep within the sun-dappled, emerald-tinted waters of the vast Indo-Pacific, where coral cities rise like ancient, silent cathedrals from the ocean floor, there exists a world teeming with life, both flamboyant and reclusive. Here, sunlight filters down through countless fathoms, breaking into shimmering shafts that dance across fields of living stone, illuminating a kaleidoscope of forms and colors. The currents, gentle as a whispered breath, weave through intricate labyrinths of branching corals carrying microscopic nourishment and the soft, murmuring echoes of the deep. It is within this vibrant, complex realm, a realm of perpetual motion and profound stillness, that one might, with a patient and discerning gaze, encounter one of the ocean's most enigmatic inhabitants, the blue-ringed octopus. This creature, often no larger than a human golf ball when fully grown, represents a fascinating paradox of the natural world. It is a being of exquisite, almost porcelain-like delicacy, yet it harbors a potency that belies its diminutive stature. When undisturbed, its skin might display a muted, earthy palette, a mottled blend of browns, greys, and sandy yellows, allowing it to dissolve effortlessly into the textured background of its rocky or coral-strewn surroundings. It becomes a phantom of the reef, a master of disguise, its soft, boneless body molding itself to the contours of crevices, and the shadows beneath overhangs a living whisper within the vibrant clamor of the underwater world. The name itself, octopus, evokes images of ancient, intelligent beings, cephalopods of remarkable dexterity and cunning, possessing a lineage stretching back through countless epochs of marine evolution. And then, blue-ringed, a descriptor that hints at the extraordinary visual spectacle that defines this particular species, a feature that transforms it from a camouflaged shadow into a breathtaking, albeit dangerous, jewel. These small octopuses are not solitary wanderers of the open ocean, but rather intimate residents of the coastal shallows, preferring the intricate shelter offered by tide pools, rocky reefs, and beds of seagrass across a wide expanse from northern Australia through Southeast Asia and stretching west towards Sri Lanka and India and east to Japan. Each specific locale lends its own subtle environmental nuances to their existence, yet their fundamental nature remains steadfast. Imagine for a moment the quiet grace of its movement. Its 8 arms, slender and flexible, propel its bulbous mantle with an almost liquid fluidity. It does not swim with the powerful thrust of a fish, but rather undulates and glides a phantom drifting through the water column. Or it might crawl with a deliberate, almost meditative pace across the rough surfaces of coral, its suckers adhering with an astonishing precision. Each arm is a marvel of biological engineering capable of independent action, yet working in concert, exploring, gripping and navigating the complex topography of its submerged habitat. It moves with such a profound quietude, a silence inherent to its aquatic existence, that it truly earns its moniker as a silent jewel of the deep. Its gaze, though often hidden, emanates from eyes that are surprisingly complex, allowing it to perceive the intricate details of its immediate world, detecting the subtle shifts in light and shadow that might betray the presence of a predator or the elusive movements of its next meal. For much of its brief life, which typically spans only a year or two, the blue-ringed octopus remains a creature of reclusion, a shy artisan of concealment. It prefers the cool, dim shelter of a discarded seashell, a forgotten glass bottle, or a narrow fissure within a rock formation, places where its small form can vanish from the prying eyes of larger marine predators such as moray eels or certain species of fish. In these hidden sanctuaries, it spends its days in quiet contemplation, emerging primarily under the cover of dusk or dawn, or perhaps during the deep tranquility of night, to hunt for small crabs, shrimp, or even tiny fish that might venture too close to its camouflage lair. Its hunting strategy is one of patience and precision, a testament to its cephalopod intelligence, lying in wait perfectly still until the opportune moment arrives for a swift, decisive strike. The allure of this creature lies not only in its exquisite, understated beauty, but also in the profound sense of mystery it carries. It is a living testament to the ocean's capacity for creating life forms that challenge our perceptions of power and vulnerability. Its small size, far from suggesting insignificance, becomes a crucial element of its mystique, underscoring the ancient adage that true strength often resides in the most unexpected places. The very act of observing such a creature, even in the mind's eye, evokes a sense of wonder, a quiet reverence for the intricate, often unseen forces that shape the natural world. When this octopus is at ease, its skin might even ripple with subtle, iridescent hues, a play of light upon its textured surface, hinting at the hidden brilliance beneath. It is a creature that seems to absorb the very essence of its surroundings, reflecting the dappled light and the muted colors of the seabed. But it is when this tranquility is disturbed, when a threat is perceived, that the blue-ringed octopus undergoes its most dramatic and unforgettable transformation. In a flash, almost instantaneously, its skin darkens, turning a deep, rich ochre, or a startling velvety brown, providing a stark canvas upon which its eponymous blue rings ignite. These rings, perhaps 24 or more, scattered across its mantle, and arms, are not merely painted-on patterns. They are chromatophores, specialized pigment cells that expand and contract with astonishing speed, revealing an underlying layer of vibrant, iridescent blue. This sudden luminous display is not a gentle shimmer, but a pulsating, almost electric glow, a vivid declaration against the darker background of its body. The rings appear to throb with an internal light, a brilliant, almost otherworldly azure that draws the eye with an undeniable force. Each ring, perfectly circular, seems to burn with an inner fire, a vivid warning that is both mesmerizing and profoundly unsettling. It is a silent language spoken not through sound, but through pure, unadulterated visual intensity, an ancient signal honed over millennia of evolutionary refinement. This transformation from a demure camouflage presence to a beacon of vibrant, flashing danger is one of nature's most compelling visual narratives, a story told in hues of deepest ocean and electric sky. These rings are not always visible, they are a deliberate, conscious activation, a sophisticated form of communication that speaks directly to the visual centers of any potential aggressor. The sheer speed with which these rings materialize from a state of near invisibility to a dazzling, pulsating spectacle is a testament to the octopus's remarkable physiological control. It is a living signal lamp broadcasting a message that is universally understood in the silent depths. Approach no further. The very sight of these rings, even for a moment, etches itself into the memory, a vivid reminder of the profound and often hidden power that resides within the ocean's most delicate forms. To truly appreciate this creature is to understand its place within the intricate web of marine life, a small but significant thread in the vast, interconnected fabric of the reef ecosystem. It is a predator, yes, but also prey, navigating its existence with a keen awareness of its surroundings, always seeking balance between its need for sustenance and its imperative for survival. Its relatively short lifespan means that each generation must master the ancient art of camouflage hunting and defense, passing on the subtle wisdom of its kind through genetic inheritance. As we gently drift through these initial observations, allowing the image of this quiet yet potent marine inhabitant to settle softly into the mind, we begin to perceive the layers of its being. From its humble camouflaged beginnings to the sudden electrifying brilliance of its warning display. The blue-ringed octopus invites a deeper contemplation of the natural world's profound intricacies. It is a creature that embodies both serene beauty and an undeniable inherent power, a silent jewel that guards its secrets with a vibrant, unforgettable flash of blue. This initial glimpse into its world merely sets the stage for a more profound exploration of its existence, its unique biology, and the remarkable mechanisms that govern its life in the dreamy ocean depths. The quiet currents of this story have just begun to carry us, a glimpse into the depths. Discovery and classification. The soft, rhythmic pulse of the ocean continues its endless conversation with the shore, a whispered story told across eons. We have glimpsed the blue-ringed octopus, a creature of quietude and sudden, vibrant warning, a small living jewel nestled within the intricate architecture of the coral reef. But how did humanity, with its terrestrial gaze and its slow, deliberate methods of understanding, come to know such a hidden marvel? How did this elusive being, often no bigger than the palm of a hand, emerge from the vast, shimmering mystery of the deep to find its place within the meticulously ordered scrolls of scientific knowledge? The journey of discovery. For many of the ocean's most reclusive inhabitants often begins not with a grand expedition, but with chance encounters with the curious eyes of local fishers or the patient observations of naturalists drawn to the boundless wonders of coastal ecosystems. For the blue-ringed octopus, its initial entry into the formal annals of science unfolded gradually, perhaps hinted at, in the lore of coastal communities long before it received its Latin designation. These communities, living in intimate proximity to the sea, would have been acutely aware of the subtle dangers and hidden beauties that populated their watery world. Their knowledge passed down through generations, often without the need for formal labels. It was during the great age of natural history exploration, a period stretching from the late 18th century well into the 19th and early 20th centuries, that Western science began to systematically catalogue the planet's biodiversity. Ships laden with eager scientists, artists, and collectors sailed into uncharted waters, their holds often filled with specimen jars, and their minds alight with the thrill of the unknown. It was within this spirit of meticulous observation and classification that the genus Hapalochlaena, the scientific grouping to which all blue-ringed octopuses belong, slowly began to materialize from the oceanic mists. The earliest formal descriptions of these creatures often involved the painstaking process of collecting specimens sometimes preserved in alcohol, then meticulously sketching and detailing their physical characteristics under the soft glow of a lantern or the filtered light of a laboratory window. Imagine a dedicated naturalist, perhaps, in a quiet study filled with the scent of brine and preserving fluids leaning over a small, mottled creature, its vibrant rings now muted by preservation, yet still hinting at the startling beauty it possessed in life. Every suckered arm, every subtle fold of its mantle, every minute detail of its eyes would have been carefully noted compared against existing knowledge, and then slowly, a new entry into the grand encyclopedia of life would begin to take form. One of the most widely recognized species, the southern blue-ringed octopus, Hapalochlaena maculosa, was formally described in 1832 by the British zoologist George Shaw. Think of the world at that time: no underwater cameras, no remotely operated vehicles, just the keen human eye, the steady hand of a sketch artist, and the intellect of a scientist piecing together the fragments of a vast, unseen mosaic. These early descriptions were not merely acts of naming, but profound attempts to understand, to categorize, and to place each new discovery within the overarching narrative of life's evolution. The maculosa in its name derived from Latin hints at its spotted or mottled appearance, a characteristic that in its quiescent state allows it to blend seamlessly with the textured seabed. Later, other species within the genus would be identified, such as Hapalochlaena lunulata, the greater blue-ringed octopus described in 1838 by Jean-René Constantin and Joseph Paul Gaimard, French naturalists on a circumnavigation voyage. The term lunulata refers to the crescent or moon-shaped appearance of its rings, a subtle distinction that further highlights the astonishing diversity even within this small group. Each discovery, each careful classification added another brushstroke to our understanding of these captivating yet perilous denizens of the Indo-Pacific. To truly comprehend the blue-ringed octopus, we must understand its place within the grand hierarchical structure of biological classification, a system that organizes all known life into progressively inclusive groups, like nested dolls. At the very broadest level, the blue-ringed octopus belongs to the kingdom Animalia, a vast realm encompassing all multicellular organisms that obtain nutrients by consuming other organisms and are generally mobile at some stage of their life cycle. Picture the sheer diversity within this kingdom, from the smallest insect to the largest whale, and understand that our small octopus shares this fundamental animal nature, a shared ancestry reaching back to the dawn of complex life. Moving deeper into this classification, we arrive at the pond phylum Mollusca. This is an ancient and incredibly diverse group, second only to arthropods in terms of the number of described species, showcasing an astonishing array of forms. Think of the slow-moving garden snail, the filter-feeding clam, the iridescent abalone, and the swift, darting squid. All are mollusks. What unites them is a shared body plan, typically featuring a soft, unsegmented body, often protected by a calcareous shell, though this has been reduced or lost in many groups, including octopuses, a muscular foot for locomotion, and a mantle which is a specialized layer of tissue that secretes the shell and encloses the visceral organs. The blue-ringed octopus, despite its lack of an external shell, carries the echoes of this ancient mollusk heritage within its soft, flexible form and its remarkable mantle. Our journey through classification then brings us to the class Cephalopoda. This is where the story truly begins to narrow and focus on the unique lineage of the octopus. The name cephalopod itself is derived from Greek, meaning head-footed, a poetic description of how their arms and tentacles emanate directly from their head region. These are the most evolutionarily advanced of all mollusks, creatures distinguished by their remarkable intelligence, highly developed nervous systems, and sophisticated sensory organs, particularly their large, complex eyes that rival those of vertebrates in their acuity. They possess a unique form of propulsion, expelling water through a funnel-like siphon, allowing for rapid, directed movement. And as we gently observed in our first chapter, cephalopods are masters of camouflage, capable of instantaneous changes in skin texture and color through the rapid expansion and contraction of specialized pigment cells called chromatophores. The blue-ringed octopus, with its dazzling warning display, is a prime example of this cephalopod mastery. Within the class Cephalopoda, we find the order Octopoda, encompassing all octopuses. These are distinguished from their cephalopod cousins, the squid and cuttlefish, by typically possessing 8 arms, as opposed to 10 appendages, often 8 arms and 2 longer tentacles, a generally rounder sac-like body, and a preference for a benthic or bottom-dwelling lifestyle. Octopuses are renowned for their incredible dexterity, their ability to squeeze through impossibly small openings, and their profound capacity for problem-solving, often observed manipulating objects or navigating complex mazes in laboratory settings. The blue-ringed octopus, with its 8 graceful arms and its habit of sheltering in crevices, perfectly embodies the characteristics of this order. Further refining our classification, we arrive at the Shriuhom, family Octopodidae. Often referred to as the true octopuses. This family includes the vast majority of commonly recognized octopus species, sharing general anatomical features and behaviors. It is a diverse family, yet bound by common evolutionary threads that have led to their success in a multitude of marine environments, from shallow reefs to the abyssal plains. And finally, we reach the specific genus Hapalochlaena. This is the exclusive domain of the blue-ringed octopuses. The name Hapalochlaena itself is derived from Greek words where hapalos means soft or tender, and chlaina refers to a cloak or mantle, a subtle nod to their soft, pliable bodies. Within this genus, scientists currently recognize several distinct species, typically 3 or 4, though ongoing research may refine this number. Each species, while sharing the iconic blue rings and potent venom, exhibits subtle differences in its size, the precise pattern and color of its rings, and its geographical distribution. For instance, Hapalochlaena maculosa. The southern blue-ringed octopus often exhibits more irregular blotchy rings, while Hapalochlaena lunulata. The greater blue-ringed octopus displays more distinct, often slightly larger circular rings. These distinctions, though subtle to the untrained eye, are crucial for scientists in understanding the evolutionary pathways and ecological roles of each unique species. This journey through discovery and classification is more than a mere listing of names. It is a profound act of understanding, a quiet meditation on the interconnectedness of all life. Each level of classification tells a part of the blue-ringed octopus's ancient story, from its fundamental animal nature to its specific adaptations as a venomous camouflaged hunter of the Indo-Pacific reefs. It allows us to appreciate the meticulous work of generations of scientists who, with patience and curiosity, have slowly, gently peeled back the layers of the ocean's mysteries, bringing creatures like the blue-ringed octopus from the shadowed depths into the clear light of human comprehension. As these scientific layers settle softly in your mind, let the quiet weight of this knowledge gently guide you further into the tranquil embrace of the deep. The canvas of warning. Anatomy and chromatophore magic. As the quiet currents of our story continue to carry us gently, drawing us deeper into the hushed realm of the blue-ringed octopus we leave behind, the chronicles of its discovery and its careful placement within the grand tree of life. Now our gaze softly settles upon the creature itself, upon the delicate yet formidable architecture of its being a living sculpture crafted by millennia of evolution. We seek to understand the very fabric of its existence, from the soft undulations of its mantle to the extraordinary cellular mechanisms that orchestrate its famed incandescent warning. Picture its form a small, soft-bodied enigma, typically no larger than a human golf ball, sometimes reaching the size of an apple in certain species. Its central body, known as the mantle, is a fluid, sac-like structure utterly devoid of internal bones, granting it an astonishing plasticity. This supple, muscular sheath protects the vital organs nestled within. The gills that extract life-giving oxygen from the cool ocean water. The heart that pulses nutrients through its body, and the complex digestive system that processes its prey. The mantle is not merely a protective casing, it is also a key player in its locomotion. Through rhythmic contractions, it draws water into the mantle cavity and then expels it forcefully through a muscular tube called the siphon or funnel, propelling the octopus through the water with a silent jet-like thrust, a graceful, almost ghostly movement that leaves scarcely a ripple in its wake. Fused directly to this pliable mantle is its head, a distinct yet seamlessly integrated part of its anatomy. And upon this head, two large, remarkably complex eyes are set like dark, polished jewels. Unlike our own eyes, these lack eyelids, remaining perpetually open to the liquid world around them, ever watchful, ever alert. These eyes are a marvel of convergent evolution, possessing a lens iris and retina that function with a precision comparable to those of vertebrates. Yet they developed independently, a testament to nature's ingenious solutions to the challenge of sight. They perceive the subtle shifts in light and shadow, the fleeting movements of potential prey, and the looming forms of predators, painting a vivid, ever-changing portrait of its submerged existence. The pupils, often a horizontal slit, grant it a wide field of vision, allowing it to scan its surroundings with an almost unsettling intensity. Radiating from beneath its head are its 8 powerful arms, each a testament to flexibility and strength, a marvel of hydrostatic skeletal support. These arms are not rigid limbs, but rather muscular hydrostats, meaning they derive their support from incompressible fluids within their tissues, allowing for an unparalleled range of motion. Each arm can bend, twist, extend, and contract independently, or work in perfect synchronicity with its fellows, enabling the octopus to grasp, explore, and manipulate its environment with astonishing dexterity. Along the underside of each arm, arranged in neat, orderly rows, are hundreds of suckers. These aren't merely passive adhesion devices. Each sucker is a sophisticated sensory organ capable of tasting and touching its surroundings. They allow the octopus to cling with immense force to rough surfaces, to gently explore the texture of a rock, or to firmly secure a struggling crab, its grip unwavering, its tactile senses gathering information about its world. Hidden within the center of its 8 arms, nestled at the base where they converge, lies one of its most critical and formidable anatomical features, its beak. This structure, reminiscent of a parrot's beak but composed of tough, chitinous material, is incredibly strong and sharp. It is used to tear through the exoskeletons of crabs and the flesh of fish, its powerful jaws capable of delivering a crushing bite. And it is through this very beak that the blue-ringed octopus delivers its potent silent weapon, its venom, into the tissues of its prey, or if provoked, into the skin of an unfortunate intruder. The beak, therefore, is not just an eating tool. It is the final, crucial component in its predatory and defensive arsenal, a small, hidden instrument of lethal precision. Now, let us turn our attention to the most visually arresting aspect of this creature, the very canvas of warning that gives it its name, its skin. This is not merely an outer covering, but a dynamic, living tapestry of cellular artistry capable of instantaneous transformation. The blue-ringed octopus, like all cephalopods, possesses an extraordinary ability to change its skin color and texture with breathtaking speed and accuracy. This remarkable feat is orchestrated by specialized pigment cells known as chromatophores, along with other structural elements that interact with light in unique ways. Imagine the skin as a multi-layered masterpiece, a biological canvas painted from within. The primary actors in its rapid color changes are the chromatophores. These are tiny, elastic sacs of pigment, primarily melanin, which produces dark browns and blacks, but also xanthine for yellows and erythrin for reds and oranges. Each sac surrounded by a ring of radial muscles. When the octopus wishes to display a particular color, it consciously contracts these muscles, pulling the pigment sac open and expanding its surface area, revealing the vibrant color within. When it wishes to hide that color, the muscles relax and the elastic sac shrinks back to a tiny, almost invisible point. This process is under direct neural control, allowing for changes that are not only rapid but also incredibly precise, often occurring in mere milliseconds. This direct link to its nervous system is a hallmark of cephalopod intelligence, allowing for sophisticated communication and camouflage. When the blue-ringed octopus is at rest or seeking to blend in, its chromatophores are typically expanded, displaying a muted, mottled pattern of browns, grays, and sandy yellows, perfectly mimicking the textured seabed, the shadows of coral, or the detritus of a rock pool. It becomes a ghost, a whisper of form and shadow. But the blue rings themselves are not generated by chromatophores. They are the work of another equally fascinating type of cell called iridophores. Unlike chromatophores, which contain pigment, iridophores are structural color cells. They contain stacks of thin reflective protein plates that interact with light scattering and reflecting specific wavelengths to produce iridescent metallic or shimmering colors. It is these iridophores arranged in precise circular patterns beneath the octopus's outer skin layers that produce the brilliant electric blue of its warning rings. These blue rings are always present, a permanent feature of its skin, but they are normally concealed by the expanded dark chromatophores overlying them. When the blue-ringed octopus perceives a threat, a truly astonishing sequence of events unfolds. First, its background skin rapidly darkens, often turning a deep velvety brown or a stark contrasting ochre. This is achieved by the maximal expansion of its dark pigment chromatophores, creating a dramatic, almost theatrical backdrop. Simultaneously, and with incredible precision, the chromatophores directly overlying the iridophore rings contract and shrink, pulling back like tiny curtains. This action instantly reveals the underlying iridophores, which then blaze forth with their characteristic iridescent, almost luminescent blue. The effect is not a static glow. the octopus can rapidly expand and contract the surrounding dark chromatophores, creating a mesmerizing, pulsating or flashing effect of the blue rings. These rings appear to throb with an internal light, an undeniable, vivid declaration of danger. Imagine the sudden, almost hypnotic burst of dozens of these electric blue circles against a darkened body, each one seeming to ignite and dim in a rhythmic, deliberate display. This rapid dynamic flashing intensifies the warning, making it impossible to ignore. It is a visual language of profound urgency, a silent scream in a world without sound. Beyond chromatophores and iridophores, other cells contribute to this living canvas. Leucophores, for instance, are cells that scatter all wavelengths of light, contributing to white or reflective areas, further enhancing the contrast of the blue rings against the dark background. And the octopus can also alter its skin texture, producing small, temporary bumps or papillae, which allow it to mimic the rough surface of rocks or coral, adding another dimension to its camouflage. These papillae are controlled by tiny muscles, allowing for instantaneous changes in its dermal landscape. In essence, the blue-ringed octopus is not merely changing color, it is orchestrating a complex, multi-layered visual symphony, a silent, urgent performance of self-preservation. Its anatomy, from the fluid grace of its mantle and arms to the intricate cellular dance of its skin, is a testament to its evolutionary success in a world fraught with both beauty and peril. As we contemplate this living canvas, allowing the imagery of its quiet grace and its sudden, vibrant warning to settle deeply within, let the quiet magic of its form gently guide your thoughts, easing them into a profound and peaceful rest. The flashing azure, decoding the danger display. The quiet currents of our underwater journey continue to carry us gently through the intricate wonders of the blue-ringed octopus. We have explored the delicate architecture of its form, from the supple undulations of its mantle to the astonishing cellular artistry of its skin. Now, our focus drifts to the very heart of its mystique, the breathtaking spectacle that names it the flashing azure, the vivid pulsating declaration of danger that transforms this quiet reef dweller into a living beacon of warning. Imagine, if you will, the serene underwater world, bathed in the muted, shifting greens and blues of filtered sunlight. Our small octopus, perhaps nestled within the crevice of a coral head, or camouflaged against a patch of sand, blends seamlessly into its surroundings. Its skin, a mottled canvas of browns and greys, whispers of its presence rather than proclaiming it. It is a master of concealment, a living shadow moving with a liquid grace that barely disturbs the water around it. This state of quietude is its preferred mode of existence, a testament to its shy, reclusive nature. It conserves energy, avoids detection, and navigates its world with a silent, almost meditative purpose. But then, a disturbance, perhaps a larger fish, a curious diver or a clumsy crab, ventures too close, infringing upon the octopus's personal space, disrupting its peaceful repose. The initial response is often subtle, a slight stiffening of its posture, a momentary pause in its gentle movements. It might attempt to retreat further into its sanctuary, or flatten its body, hoping to become even more invisible. This is its first line of defense, avoidance, discretion, the silent plea to be left undisturbed. It is a creature that does not seek confrontation, preferring the quiet anonymity of its hidden life. However, if the perceived threat persists, if the intrusion continues, a remarkable physiological transformation begins to unfold, one of nature's most compelling visual alarms. The first noticeable change is often a rapid darkening of its entire body. The muted browns and greys deepen into a rich, velvety ochre, or a stark, almost blackish brown. This sudden dramatic shift in its background coloration is orchestrated by the rapid expansion of its dark chromatophores, the pigment sacs we gently explored in our previous chapter. This darkening creates a stark, dramatic canvas, a profound contrast that is essential for the next even more startling act of its display. Against this newly darkened backdrop, the true spectacle ignites. The approximately 50 to 60 iridescent blue rings, which lie dormant and concealed beneath the expanded dark chromatophores, are now revealed in a breathtaking flash. This revelation is achieved by the simultaneous contraction of the chromatophores directly overlying these rings. Like tiny, precisely controlled curtains, these dark cells retract, pulling back to unveil the shimmering structural blue of the iridophores beneath. The effect is instantaneous, almost magical, transforming the octopus from a camouflaged shadow into a beacon of pulsating light. These rings are not merely static circles of blue. They appear to throb to pulse with an internal electric energy. The surrounding dark chromatophores can rapidly expand and contract around the iridophores, creating a mesmerizing rhythmic flicker, an almost hypnotic undulation of light and shadow. The blue itself is not a flat color. It is an iridescent, almost luminescent azure, a vivid, almost neon quality that seems to burn with an inner fire. Each ring, perfectly circular or perhaps slightly crescent-shaped depending on the species, stands out with an undeniable intensity, drawing the eye with a potent, almost magnetic force. The sheer speed of this transformation from near invisibility to a dazzling, pulsating warning is a testament to the octopus's highly evolved nervous system and its remarkable control over its dermal landscape. This flamboyant display is a classic example of epicemitism, an evolutionary strategy where an animal advertises its unpalatability or danger to potential predators through a conspicuous signal. In the terrestrial world, we see aposematism in the vibrant stripes of a wasp, the bold patterns of a poisonous frog, or the vivid colors of a toxic butterfly. In the marine realm, the blue-ringed octopus's flashing azure rings serve the same powerful purpose, a clear, unambiguous declaration to any would-be aggressor: I am dangerous. Do not touch. The effectiveness of this display lies in its universality. In the silent language of nature, bright contrasting colors, especially when combined with a sudden dynamic presentation, are often understood as a signal of toxicity or potent defense. A predator, whether a fish, an eel, or even a curious human, is likely to possess an innate or learned aversion to such striking visual cues. The brain of a potential threat registers this vivid pattern, associating it with previous negative experiences or an instinctive understanding of danger. The message is simple, direct, and profoundly effective. The risk of engaging this creature far outweighs any potential reward. Scientists believe that the specific blue hue combined with the pulsating effect is particularly potent because it creates a strong visual contrast against the darkened body, making the rings highly visible even in the dim light of the reef or during twilight hours when the octopus might be more active. The dynamic flashing further enhances its impact, making it more difficult for a predator to ignore or misinterpret. It's not just a warning, it's an urgent, flashing exclamation mark. The blue-ringed octopus does not employ this display lightly. It is an energy-intensive act requiring rapid muscular contractions and precise neural control. Furthermore, by displaying its rings, the octopus temporarily sacrifices its camouflage, making itself highly visible in an environment where blending in is often key to survival. Therefore, the flashing azure is a last resort, a final, unequivocal warning before the octopus considers deploying its ultimate defense, its potent venom. It is a declaration that says, "I have given you every chance to retreat. My next action will be irreversible." When the octopus unfurls its arms and mantle to reveal the full glory of its pulsating rings. It is not merely a visual display, it is a full-body commitment to its defensive posture. Its body might even flatten, or it might arch its back, presenting the maximum surface area of its warning coloration to the perceived threat. This combination of episomatic coloration and defensive body language reinforces the message, leaving little room for doubt about its intent or its capabilities. The profound beauty of this danger display, the almost hypnotic quality of the flashing azure, serves as a poignant reminder of the intricate, often perilous wonders that lie hidden beneath the ocean's surface. It speaks to the ancient wisdom of evolution, to the countless generations that refined this silent language of warning, ensuring the survival of a small, delicate creature, armed with an invisible yet profoundly lethal power. As the imagery of these pulsating blue rings gently fades from your mind's eye, allow the quiet understanding of their potent message to guide you further into the tranquil depths of your own restful sleep. Hidden lives in the reef, habitat, and daily rhythms. The vibrant, pulsating azure of the warning display, a fleeting beacon in the watery expanse, now gently recedes from our mind's eye, dissolving back into the quiet depths from which it emerged. As the last echoes of that visual alarm softly fade, we turn our gaze toward the more enduring rhythms of the blue-ringed octopus's existence, to the hidden lives it leads within the intricate architecture of its chosen home. For most of its days and nights, this creature is not a flashing signal of danger, but a master of stealth, a reclusive inhabitant of the vast, sun-dappled and shadow-drenched Indo-Pacific oceans. Imagine the sheer breadth of its realm stretching across a colossal expanse of our planet's surface. From the northern coasts of Australia, where the Great Barrier Reef unfurls its kaleidoscopic wonders, through the myriad islands and archipelagos of Southeast Asia, the Philippines, Indonesia, Malaysia, and extending westward to the shores of Sri Lanka and India, then northward to the temperate waters off Japan, this small octopus makes its home. It is a vast, interconnected network of marine ecosystems, each with its own subtle variations in temperature, salinity, and current, yet all offering the crucial elements this species requires for its survival. Within this expansive geographical range, the blue-ringed octopus is not a creature of the open ocean drifting aimlessly with the currents. Instead, it is a benthic dweller, meaning it prefers to live intimately connected to the seabed, seeking out the complex topography of coastal shallows. Its preferred habitats are a mosaic of rocky reefs, where ancient stones are encrusted with a living tapestry of algae and anemones. Vibrant coral reefs, rising like submerged cities carved from calcium carbonate. Tranquil seagrass beds where long green blades sway gently with the ebb and flow of the tides. And even the less glamorous yet equally vital rubble zones, areas strewn with broken coral fragments, discarded shells, and the detritus of the ocean floor. The absolute cornerstone of its existence within these habitats is the availability of secure camouflaged shelter. This octopus does not build elaborate burrows or construct intricate nests in the manner of some other marine creatures. Instead, it is an opportunistic occupant, a resourceful seeker of existing voids and protective crevices. Its soft, boneless body is an extraordinary advantage in this regard, allowing it to squeeze through impossibly small openings, to flow like liquid into tight fissures in rocks, or to disappear into the hollow interior of a discarded seashell. Picture it, its body contracting and elongating its 8 flexible arms, probing and guiding, until it has seamlessly melded with the contours of its chosen sanctuary. These chosen dens are often humble yet perfectly functional. A cracked beer bottle half buried in the sand, a forgotten tin can slowly corroding and becoming part of the seabed. A small, empty clamshell, its pearly interior now a private chamber. A narrow, winding tunnel within a porous piece of coral. All these can serve as its temporary abode. The critical requirement is that the shelter provides concealment from the keen eyes of predators such as moray eels, various species of fish, and even some seabirds during low tide. It also offers a safe haven from strong currents and the relentless surge of waves, allowing the octopus to conserve its energy and remain undisturbed. Its daily rhythms are largely dictated by the sun's journey across the sky, or more precisely, its absence. The blue-ringed octopus is primarily a crepuscular and nocturnal hunter. This means it is most active during the twilight hours of dawn and dusk, when the light is dim and dappled, and throughout the deep tranquility of night. During these periods, many of its own predators are less active, and its preferred prey, small crabs, shrimp, and tiny fish, are often more venturesome, emerging from their own hiding places to feed. Imagine the reef at dusk, as the vibrant colors of day begin to soften, melting into hues of deep purple and indigo. Our octopus, which has spent the daylight hours in a state of quiet repose within its chosen den, now begins to stir. Its eyes, those complex marvels, become acutely attuned to the diminishing light, allowing it to perceive the subtle movements of its awakening world. It might slowly, cautiously emerge from its shelter, its body still cloaked in the muted, camouflaged patterns that allow it to blend almost perfectly with the textured seabed. It does not rush, but rather moves with a deliberate, almost meditative pace. Its 8 arms gliding over rocks and sand, its suckers testing the surfaces, gathering information about its immediate surroundings. Its hunting strategy is one of profound patience and calculated precision. It is an ambush predator. A master of the wait-and-strike technique, it might find a strategic vantage point, a slightly elevated rock or a shadowy overhang, and settle there. Its body flattened, its skin patterns shifting to perfectly mimic the texture and color of its perch. It becomes a living part of the landscape, its presence virtually undetectable to an unwary crab scuttling past, or a shrimp foraging for detritus. Its eyes, however, remain keenly focused, tracking the subtle vibrations in the water, the almost imperceptible movements that betray the presence of its next meal. When a suitable prey item comes within striking distance, the octopus erupts from its stillness with astonishing speed. Its arms, previously held close to its body, extend with lightning quickness, enveloping the unfortunate victim. The powerful suckers on its arms latch on with an unwavering grip, securing the struggling prey. Then, with its sharp, chitinous beak located in the center of its arms, it delivers a precise bite, injecting its potent neurotoxic venom. The venom, as we shall explore in greater detail, acts swiftly paralyzing the prey, rendering it helpless, allowing the octopus to consume its meal in relative peace and safety within its den or a secluded spot. Movement for the blue-ringed octopus is a blend of graceful crawling and efficient jet propulsion. When exploring its immediate vicinity or moving slowly across the seabed, it will often walk on its arms, using the suckers to grip and pull itself forward. This method allows for quiet, controlled movement, perfect for stealthy hunting or careful navigation of complex terrain. However, if it needs to escape quickly from a perceived threat or to cover a greater distance, it employs its jet propulsion system. By rapidly expelling water from its siphon, it can dart through the water with surprising speed, a sudden burst of motion that can leave a predator disoriented. Throughout its brief lifespan, typically spanning only a year or two, the blue-ringed octopus maintains a largely solitary existence. Interactions with other octopuses are generally limited to mating a fleeting encounter that ensures the continuation of its species. This solitary nature is common among many octopus species, reflecting their predatory lifestyle and their need for personal territory and shelter. Each octopus is a self-sufficient hunter, a discreet resident of its own small hidden world within the vastness of the reef. The environmental conditions of its habitat also shape its daily rhythms. In tide pools, for instance, where water levels can fluctuate dramatically with the ebb and flow of the tides, the octopus must be adept at finding shelters that will remain submerged, or at least provide adequate moisture during low tide. It might retreat into deeper rock pools or bury itself partially in the sand, waiting patiently for the return of the life-giving water. Its resilience and adaptability to these changing conditions are a testament to its evolutionary fitness, allowing it to thrive in environments that might be challenging for less adaptable creatures. As the quiet understanding of these hidden lives, these patient rhythms of hunting and hiding, gently settles within your mind, allow yourself to drift further into the tranquil depths. The small camouflaged form of the blue-ringed octopus, moving with its liquid grace, becomes a silent guide through the slumbering reef, inviting your own thoughts to find a gentle, peaceful repose within the quiet embrace of the deep. The alchemist within. Venom production and glands. As the gentle currents of our narrative continue their unhurried journey, guiding us away from the hidden lives and quiet rhythms of the blue-ringed octopus, within its reef sanctuary, we now turn our focus inward. We delve beneath the exquisite canvas of its skin, beyond the mesmerizing flash of its azure rings, to explore the profound alchemy that takes place within its delicate, soft-bodied form. Here, in a secret, silent laboratory of living tissue, lies the source of its formidable power. The venom, a potent elixir that transforms this small, unassuming creature into one of the ocean's most respected and feared inhabitants. The very concept of venom, a complex biological cocktail designed for defense or predation, often evokes images of fangs or stings. Yet in the blue-ringed octopus, this lethal capability is woven subtly into its very being, a testament to nature's boundless ingenuity. The production and storage of this potent substance are not a simple, straightforward process, but rather a remarkable story of biological cooperation, a symbiotic relationship that has evolved over countless millennia. At the heart of this internal chemistry are specialized structures known as the major, posterior salivary glands. These are not merely glands that moisten food. They are sophisticated biological factories nestled deep within the octopus's head, surrounding its esophagus. Imagine them as small, intricate organs, perhaps resembling clusters of microscopic grapes, bathed in the internal fluids of the octopus, quietly, ceaselessly working. It is within the intricate folded tissues of these glands that the primary and most lethal component of the blue-ringed octopus's venom is synthesized and stored. This primary toxin is called tetrodotoxin, or TTX for short. It is a molecule of astonishing potency, a non-protein neurotoxin that is capable of disrupting the most fundamental processes of life in its victims. What makes the blue-ringed octopus's relationship with this toxin so utterly fascinating is that the octopus itself does not produce TTX. This is a crucial, almost poetic detail in its biological story. Instead, scientists have discovered that the tetrodotoxin is produced by specific strains of symbiotic bacteria that reside within the octopus's salivary glands. Consider the concept of symbiosis, a profound partnership between different organisms living in close association, often to their mutual benefit. In this particular case, the octopus provides a warm, protected, nutrient-rich environment within its salivary glands, a perfect habitat for these microscopic bacterial alchemists. In return, the bacteria tirelessly synthesize the tetrodotoxin, a chemical weapon that confers an unparalleled advantage upon their cephalopod host. It is a silent contract, an ancient agreement forged in the crucible of evolution, ensuring the survival and predatory success of the octopus. While the exact species of bacteria responsible can vary and is still a subject of ongoing scientific investigation, research points to certain marine bacteria, often belonging to groups like Vibrio or Pseudomonas, as the primary producers. Imagine these minuscule organisms invisible to the naked eye, diligently performing their biochemical tasks within the confines of the octopus's glands. Each bacterium is a tiny self-contained factory equipped with the genetic blueprints and enzymatic machinery required to assemble the complex tetrodotoxin molecule atom by atom from simpler precursors. It is a process of extraordinary precision yielding a substance of immense power. The tetrodotoxin, once synthesized by these bacterial partners, accumulates within the cells of the posterior salivary glands. It is stored there in concentrated form, a ready reserve for when the octopus needs to subdue prey or defend itself. The glands are thus not just sites of production, but also secure reservoirs holding enough of this potent compound to paralyze and kill far larger creatures than the octopus itself. Scientists theorize that the octopus has evolved specific mechanisms to tolerate the presence of TTX within its own tissues, preventing it from succumbing to its own lethal weapon. This internal immunity is another marvel of biological adaptation, allowing the host to wield a toxin that would be fatal to most other animals. Beyond tetrodotoxin, the venom of the blue-ringed octopus is not a monolithic substance. While TTX is undeniably the star player contributing the primary neurotoxic effects, octopus venom in general is often a complex cocktail of various biologically active compounds. These can include proteolytic enzymes which help to break down the tissues of prey, aiding in digestion even before the meal is fully consumed. There might also be other proteins or peptides that contribute to paralysis, or even compounds that facilitate the spread of the venom through the victim's body. These additional components, while perhaps less acutely lethal than TTX, work in concert to enhance the overall effectiveness of the venom, ensuring rapid incapacitation of prey and a robust defense mechanism. The delivery system for this potent venom is as precise as its production. As we gently observed in a previous chapter, the blue-ringed octopus possesses a sharp, chitinous beak nestled at the center of its 8 arms. This beak is not just for tearing, it is the final conduit for the venom. When the octopus bites, the venom stored in its posterior salivary glands is channeled through ducts that lead directly to the tips of its beak or into specialized venom papillae located near the beak. With a swift, almost imperceptible puncture, the venom is injected deep into the tissues of its victim. The process is quick, efficient, and often goes unnoticed by the recipient until the insidious effects of the neurotoxin begin to manifest. The evolutionary advantage of this symbiotic venom production is profound. For a small, soft-bodied creature like the blue-ringed octopus, which lacks the formidable claws or powerful jaws of larger predators, an extremely potent, fast-acting venom is an essential tool for survival. It allows the octopus to quickly subdue crabs and shrimp, which are often encased in tough exoskeletons and can be difficult to handle. More critically, it provides a powerful deterrent against predators that might otherwise view it as an easy meal. The sheer lethality of TTX means that even a small amount can be devastating, making the octopus a highly respected, if not entirely avoided, resident of the reef. Consider the energy efficiency of this arrangement. The octopus does not expend its own metabolic resources to synthesize the complex TTX molecule. Instead, it delegates this demanding biochemical task to its bacterial partners. This frees up the octopus's energy for other vital functions: camouflage, hunting, reproduction, and the meticulous maintenance of its highly intelligent nervous system. It is a masterful example of biological outsourcing, a strategy that has allowed the blue-ringed octopus to thrive in a competitive and often dangerous marine environment. The mystery of how this symbiotic relationship first arose, how the bacteria came to inhabit the glands, and how the octopus developed its immunity to TTX remains a subject of ongoing scientific inquiry. Scientists believe it likely evolved over vast stretches of time, with incremental adaptations on both sides, slowly forging this remarkable and deadly partnership. It is a delicate balance, a testament to the intricate dance of life and chemistry that underpins the natural world. As we gently absorb the quiet wonder of this internal alchemy, allowing the image of these microscopic bacterial factories ceaselessly producing their potent elixir within the hidden depths of the octopus's glands to settle softly in our minds, let the profound intricacy of life's processes guide you deeper. The silent, ceaseless work of these tiny alchemists within the blue-ringed octopus becomes a soothing rhythm, easing your thoughts into a tranquil, restful state. The molecular key, unraveling the neurotoxin. The quiet hum of the ocean deep, a ceaseless ancient lullaby, continues to enfold us as we drift further into the intricate world of the blue-ringed octopus. We have gently unveiled the alchemical secrets nestled within its posterior salivary glands, witnessing the silent, ceaseless work of symbiotic bacteria that craft its potent weapon. Now our journey takes us even deeper to the very heart of this power, to the molecular key that unlocks such devastating effects. Tetrodotoxin, a compound of profound biological significance, a silent architect of stillness. Imagine a molecule exquisitely crafted not by the octopus itself, but by its microscopic partners, a complex arrangement of atoms forming a structure that is both small and extraordinarily stable. Tetrodotoxin, often referred to simply as TTX, is a non-protein neurotoxin. This distinction is important. Unlike many animal venoms, which are large, fragile proteins, TTX is a relatively small organic compound. Its compact, robust nature allows it to persist to maintain its deadly form, even under varying environmental conditions, making it a particularly formidable biological agent. It is colorless, odorless, and tasteless, a phantom of lethality, offering no sensory warning of its presence until its insidious work has already begun. To truly comprehend the profound impact of TTX, we must gently descend into the microscopic landscape of our own nervous system, into the very fabric of how our bodies communicate. Every thought, every breath, every gentle movement of a muscle is orchestrated by a delicate dance of electrical signals transmitted along nerve cells. These signals, known as action potentials, are generated by the precise rhythmic opening and closing of tiny specialized gates embedded within the membrane of every nerve and muscle cell. These gates are called voltage-gated sodium channels. Picture these sodium channels as minuscule, exquisitely sensitive doorways studded along the length of a nerve fiber, awaiting their specific cue. When a nerve impulse arrives, a subtle shift in the electrical charge across the cell. Membrane occurs a whisper of energy. This delicate electrical change acts as a signal, gently coaxing these sodium channel doorways to swing open. When they open, a rush of positively charged sodium ions, tiny energetic particles, flows from the outside of the cell to the inside. This sudden influx of positive charge is the very spark, the electrical pulse, that propagates the nerve signal forward like a wave moving across a tranquil pond. After this momentary rush, the sodium channels swiftly close, preparing for the next signal, maintaining the delicate balance that allows for continuous communication. Now, envision the tetrodotoxin molecule as a perfectly shaped yet utterly malicious key. This molecular key possesses an uncanny affinity for these voltage-gated sodium channels. It does not destroy them, nor does it permanently alter their structure in a crude way. Instead, TTX binds with astonishing precision and tenacity to the outer opening of these crucial sodium channels. It acts like a finely tuned plug, fitting snugly, almost invisibly, into the very entrance of the channel, physically obstructing the pathway. When TTX binds to these channels, it effectively jams them shut, preventing them from opening regardless of the electrical signals attempting to coax them. The sodium ions essential for generating nerve impulses are utterly blocked from flowing into the cell. The delicate electrical dance ceases. The spark cannot ignite. The wave cannot propagate. It is as if a silent, invisible hand has reached into the intricate circuitry of the body and with a single, precise movement pulled the master switch. The consequences of this molecular blockade are profound and widespread, affecting every system that relies on nerve impulse transmission. First, the motor nerves, those vital pathways that carry commands from the brain to the muscles, begin to falter. The muscles deprived of their electrical instructions can no longer contract. This leads to a progressive, insidious paralysis. It often begins subtly, a gentle tingling or numbness in the lips and tongue, perhaps in the fingertips and toes, a creeping sensation of unreality. This might then progress to a weakness in the limbs, a feeling of heaviness, as if gravity itself has suddenly intensified its embrace. The muscles, once responsive and strong, become unresponsive, limp, refusing to obey the will of the mind. As the toxin spreads and its effects deepen, the larger, more critical muscle groups begin to succumb. The muscles responsible for movement of the arms and legs become profoundly weak, then utterly flaccid. But the most terrifying and life-threatening effect of TTX is its impact on the gluteus respiratory muscles, particularly the diaphragm. The diaphragm, a dome-shaped muscle beneath the lungs, is the primary engine of breathing, contracting rhythmically to draw air into our bodies. When the nerves controlling the diaphragm are blocked by TTX, this vital muscle ceases its rhythmic contractions. The chest, once rising and falling with comforting regularity, becomes still. The breath, that most fundamental of life's processes, slowly, inexorably fades. Respiratory arrest, the cessation of breathing, is the ultimate cause of death from tetrodotoxin poisoning. What makes this paralysis particularly harrowing is that in many cases the victim's consciousness often remains perfectly intact. The tetrodotoxin does not readily cross the blood-brain barrier, the protective membrane that shields the brain from many circulating toxins. This means that the mind, the awareness, the capacity for thought and sensation can remain clear even as the body becomes a silent, unresponsive vessel. Imagine the profound terror, the utter helplessness of being fully aware, fully conscious, trapped within a body that can no longer move, no longer speak, no longer even breathe on its own. It is a silence deeper than any ocean, a stillness more absolute than any calm. The potency of tetrodotoxin is almost beyond comprehension. It is considered one of the most toxic non-protein substances known to science. For humans, the lethal dose can be incredibly small, measured in micrograms, mere millionths of a gram. A quantity no larger than a few grains of salt can be enough to be fatal. To put this into perspective, TTX is estimated to be many thousands of times more potent than cyanide, a substance already synonymous with swift and deadly poison. The blue-ringed octopus, small as it is, carries enough of this molecular key within its salivary glands to potentially kill dozens of adult humans. This chilling fact underscores the profound danger contained within its delicate form, a power inversely proportional to its size. It is also fascinating to note that the blue-ringed octopus is not alone in possessing this formidable toxin. Tetrodotoxin is found in a remarkable array of other marine and terrestrial animals, a testament to its evolutionary effectiveness. The most famous example is perhaps the pufferfish, particularly the species consumed as fugu in Japan, where improperly prepared fish can lead to fatal poisonings. Certain species of newts in North America, such as the rough-skinned newt, also harbor TTX in their skin as a defense mechanism. Even some species of frogs and starfish have been found to contain this neurotoxin. The common thread among these diverse creatures is the theory that the TTX is not produced by the animals themselves, but rather by symbiotic bacteria that they acquire from their diet or environment, much like the blue-ringed octopus. This widespread presence of TTX, always linked to microbial partners, reinforces the elegance of this ancient bacterial alchemy. As the intricate details of tetrodotoxin's molecular mechanism and its profound effects gently settle within your thoughts, allow the quiet understanding of its power to guide you further into the tranquil depths of your own rest. The silent, insidious work of this molecular key, disrupting the very flow of life's signals, becomes a profound metaphor for the quiet stillness that now begins to embrace your weary mind. The body's quiet shutdown. Effects of envenomation. The gentle, ceaseless murmur of the ocean continues its soft embrace, guiding us further into the profound intricacies of the blue-ringed octopus. We have explored the silent alchemy within its glands and unraveled the molecular blueprint of tetrodotoxin, understanding how this potent compound silences the vital channels of our nervous system. Now, with a quiet solemnity, we turn our attention to the observable, harrowing journey that unfolds when this invisible weapon is deployed, when the body with an insidious grace begins its quiet shutdown, following an envenomation. Imagine, if you will, a moment of fleeting contact, perhaps a curious hand inadvertently brushing against a camouflaged form, or a foot accidentally stepping upon a creature hidden within the sand. The bite itself, often delivered by the small, sharp beak of the octopus, can be surprisingly subtle, almost imperceptible. It might feel like a minor pinprick, a gentle scratch, or even go entirely unnoticed amidst the sensory richness of the marine environment. There is often no immediate searing pain, no dramatic alarm bell to herald the injection of a substance of such profound lethality. This initial lack of acute discomfort is perhaps one of the most treacherous aspects of blue-ringed octopus envenomation, allowing precious moments to slip away before the true danger is recognized. A tiny, almost insignificant puncture mark, perhaps just two faint red dots, might be the only visible evidence easily dismissed as a minor abrasion. Within minutes, sometimes as few as 5, sometimes stretching to perhaps an hour, the first subtle whispers of the toxin's work begin to manifest. A creeping numbness, a tingling sensation might begin to unfurl around the bite site, a localized dampening of sensation, as if the very nerve endings are slowly being lulled into a deep slumber. This initial paresthesia, a strange prickling feeling, then gently, insidiously begins to spread. It might extend from the site of contact up the limb towards the torso, a silent tide of sensory dullness. But the most critical early signs often manifest around the mouth and face. A peculiar tingling of the lips, a strange heavy sensation on the tongue, as if it has become slightly swollen or unresponsive. This oral numbness can then extend to the entire facial area, a mask of dullness settling over the features. Speech might become subtly slurred, the words forming with a newfound effort, the articulation becoming imprecise, a condition known as dysarthria. The tongue, once agile and quick, feels sluggish, unresponsive to the commands of the brain. Simultaneously, swallowing can become increasingly difficult, a sensation of constriction in the throat, a profound challenge to perform an act that is normally effortless and automatic. This dysphagia, the struggle to ingest even one's own saliva, marks a significant progression in the toxin's reach. As these initial sensory effects deepen a general feeling of malaise, a profound weakness might begin to envelop the body. The limbs, once light and responsive, might feel heavy, as if weighted down by an unseen force. A wave of dizziness, a subtle disorientation might wash over the mind, accompanied by a growing sense of nausea. Though overt vomiting is not always a consistent symptom, the head might ache with a dull, throbbing pressure, a silent protest from a system under siege. The insidious nature of tetrodotoxin means that this progression is often painless, or at least devoid of the agonizing burning pain associated with many other venoms. It is a quiet, creeping paralysis, a gradual silencing of the body's electrical symphony, rather than a violent disruption. This very lack of pain can be deceiving, lulling both the victim and those around them into a false sense of security, delaying the urgent need for medical intervention. Within a relatively short span, often between 1 to 4 hours, depending on the amount of venom injected and the individual's physiological response, the paralysis begins its relentless march across the entire voluntary muscular system. The weakness in the limbs deepens, transforming into profound, flaccid paralysis. The arms and legs, once capable of powerful movement, become limp, unresponsive, refusing to lift or grasp. The muscles that control the delicate movements of the eyes might also be affected, leading to difficulty focusing, a blurring of vision, or even an inability to move the eyeballs, a condition known as ophthalmoplegia. The reflexes, those automatic responses that protect our bodies, begin to diminish, then vanish entirely as the nerve pathways are effectively severed by the molecular blockade. The most terrifying and life-threatening consequence of this spreading paralysis is its inevitable impact on the respiratory system. The muscles responsible for breathing, particularly the diaphragm, that crucial dome-shaped muscle beneath the lungs, begin to weaken. The rhythmic contraction and relaxation that draws life-giving air into the lungs becomes shallow-strained and increasingly labored. The chest, which once rose and fell with comforting regularity, now struggles its movements, becoming minimal and desperate. This is the moment of profound crisis where the body's quiet shutdown reaches its most critical juncture. As the respiratory muscles succumb completely to the toxin's grip, the ability to breathe independently ceases. The diaphragm paralyzed and still can no longer draw air into the lungs. This leads to respiratory arrest, a cessation of breathing. Without the constant exchange of oxygen and carbon dioxide, the body rapidly begins to suffer from hypoxia, a severe deprivation of oxygen. The skin might take on a bluish tinge, especially around the lips and fingertips, a chilling sign of cyanosis as oxygen-depleted blood circulates through the body. The brain, exquisitely sensitive to oxygen starvation, begins to feel the profound effects, though remarkably consciousness can persist for a terrifyingly long period. This preservation of consciousness, even as the body becomes utterly paralyzed, is perhaps the most harrowing aspect of tetrodotoxin poisoning. The toxin primarily affects the peripheral nervous system, the nerves that connect the brain and spinal cord to the rest of the body. It does not readily penetrate the blood-brain barrier, the protective membrane that shields the central nervous system. This means that for a significant duration, the victim can remain fully aware, fully conscious, trapped within their own unresponsive body. They can hear, they can see, they can feel the creeping paralysis. They can understand the unfolding tragedy, but they cannot move a muscle, cannot speak a word, cannot even signal their distress. Their eyes might remain open, fixed, perhaps dilated, reflecting a mind that is acutely present, yet utterly imprisoned. In the final, desperate stages, if breathing is not artificially supported, the severe and prolonged lack of oxygen will eventually lead to cardiac arrest. While tetrodotoxin does not directly target the heart muscle, in the same way it paralyzes skeletal muscles, the profound oxygen deprivation eventually overwhelms the cardiovascular system. The heart, weakened by hypoxia, will eventually cease its pumping action, bringing an end to the silent, terrifying ordeal. The entire progression from the subtle initial tingling to complete respiratory arrest can unfold with a chilling swiftness, sometimes within as little as 30 minutes to an hour in severe cases, though often it stretches to several hours. This rapid onset and the unique nature of the paralysis, coupled with the preserved consciousness, make blue-ringed octopus envenomation one of the most uniquely terrifying and medically challenging toxicological emergencies. It is a testament to the profound silent power that nature can pack into the most unassuming forms, a reminder of the delicate line between life and the body's quiet, insidious shutdown. As the somber understanding of these physiological events gently settles within your mind, let the quiet weight of this knowledge guide your own awareness into a deeper, more peaceful rest, a race against time. Medical response and life support. The quiet, creeping shutdown of the body, a silent and profound consequence of tetrodotoxin's insidious work, now gently draws our attention to the urgent, unwavering efforts made in the face of such a unique biological challenge. As the last echoes of that terrifying paralysis softly settle within our understanding, we turn to the realm of human intervention, to the race against time that unfolds when a life hangs in the delicate balance between the toxin's relentless grip and the meticulous application of medical science. When an envenomation by a blue-ringed octopus is suspected, every single moment becomes precious, each passing minute, a critical component in the unfolding drama of survival. The immediate on-site response is paramount, a crucial first line of defense designed to slow the insidious spread of the toxin. The recommended first aid, particularly in regions where these octopuses are found, is the application of a pressure immobilization bandage. Imagine a firm, wide bandage, perhaps an elastic crepe bandage wrapped snugly around the affected limb, starting from the bite site and extending upwards towards the torso. This is not a tourniquet which aims to cut off all blood flow, but rather a steady, even pressure applied over a broad area. The principle behind this technique is elegantly simple, yet profoundly effective. The venom, once injected, travels through the lymphatic system, a network of delicate vessels that carry fluid and immune cells throughout the body, eventually draining into the bloodstream. Unlike the rapid flow of blood in arteries and veins, the lymphatic fluid moves much more slowly, propelled by muscle contractions. By applying firm pressure, the flow of this lymphatic fluid is gently yet significantly impeded. It is like gently squeezing a sponge, slowing the release of its contents. This delay in the lymphatic absorption of the toxin buys precious time, slowing its journey towards the central nervous system, and critically towards the vital respiratory muscles. Simultaneously, the affected limb should be immobilized, kept as still as possible to further minimize muscle movement and thus reduce the pumping action that aids lymphatic circulation. Every subtle movement, every twitch, can inadvertently hasten the toxin's journey. Following this crucial initial first aid, the most urgent priority is the swift transportation of the victim to the nearest medical facility equipped to handle such a severe neurological emergency. This often means a hospital with an intensive care unit capable of providing advanced life support. The journey itself can be fraught with anxiety, a silent race against the clock, as the effects of the venom may continue to progress during transit. Paramedics and emergency responders are trained to monitor the victim's breathing, closely ready to initiate artificial respiration if the paralysis begins to affect the vital muscles of the diaphragm and chest. They listen for the subtle changes in breath, the faint whispers of struggle, knowing that the window for intervention is narrow and unforgiving. Upon arrival at the hospital, a team of medical professionals immediately springs into action. Their initial assessment is rapid and focused, seeking to determine the extent of the paralysis and, most critically, the integrity of the victim's respiratory function. They will meticulously check vital signs, heart rate, blood pressure, oxygen saturation levels in the blood. Neurological assessments are performed, gently testing reflexes, muscle strength, and the ability to move the eyes and facial muscles. These initial observations provide a crucial baseline, a snapshot of the toxin's current grip, informing the immediate course of action. The cornerstone of treatment for blue-ringed octopus envenomation, indeed, for any severe tetrodotoxin poisoning, is aggressive and prolonged respiratory support. This is the single most critical intervention, the very essence of saving a life from this particular toxin. As we explored, tetrodotoxin does not directly destroy nerve cells or muscle tissue. It merely blocks the sodium channels, temporarily silencing the electrical signals. If the victim can be kept breathing and oxygenated through the critical period while the toxin is slowly metabolized and excreted by the body, then a full recovery is possible. The first step in establishing respiratory support is often intubation. Imagine a soft, flexible breathing tube, gently yet firmly guided through the mouth and into the trachea, the main airway leading to the lungs. This procedure is performed with immense care, often requiring sedation to ensure the patient's comfort and cooperation, although in cases of profound paralysis, sedation might be less necessary as the patient is unable to resist. Once the tube is securely in place, it is connected to a mechanical ventilator. a sophisticated machine that takes over the arduous life-sustaining work of breathing. The ventilator becomes the patient's lungs, rhythmically pushing air rich with oxygen into the lungs, and then allowing carbon dioxide to be exhaled. It breathes for the patient, ensuring a constant and adequate supply of oxygen to every cell in the body, particularly the brain, which, as we remember, remains largely conscious and aware throughout this ordeal. The settings on the ventilator are meticulously calibrated by doctors and respiratory therapists, adjusting the rate of breaths, the volume of air, and the oxygen concentration to precisely meet the patient's physiological needs. This period of mechanical ventilation can be prolonged, often lasting for many hours, sometimes even days, depending on the severity of the envenomation and the individual's metabolic rate. During this time, the patient's condition is under continuous vigilant monitoring. Electrocardiograms, or ECGs, track the heart's electrical activity, searching for any subtle irregularities. Blood pressure is measured frequently, ensuring adequate circulation. Oxygen saturation levels are constantly displayed, a numerical reassurance that the body is receiving the oxygen it so desperately needs. Blood tests are regularly performed, assessing electrolyte balance, kidney function, and the overall physiological response to the toxin. Beyond the immediate respiratory support, comprehensive, supportive care is vital. Intravenous fluids are administered to maintain hydration and to help the kidneys excrete the toxin. A urinary catheter is often placed to monitor urine output, a crucial indicator of kidney function. Eye drops or ointments may be used to protect the eyes, which remain open and vulnerable due to the paralysis of the eyelids. Careful attention is paid to skin care, repositioning the patient regularly to prevent pressure sores, a common complication of prolonged immobility. Nutrition is provided through a feeding tube, ensuring the body receives the energy required for healing and recovery. The psychological dimension of this experience is often overlooked, yet it is profoundly harrowing. To be fully conscious, to hear, to see, to think, but to be utterly incapable of movement, speech, or even a subtle gesture of distress is a terrifying ordeal. Medical staff are keenly aware of this unique horror. While direct communication is impossible, they strive to maintain a calm, reassuring presence, speaking gently to the patient, explaining procedures, and offering what comfort they can, knowing that the mind within the paralyzed body is acutely listening and processing every sound, every touch. The recovery, when it begins, is often gradual, a slow reawakening of the nervous system. As the body slowly metabolizes and eliminates the tetrodotoxin, the sodium channels gradually become unblocked, allowing nerve impulses to once again flow. The first signs of recovery might be a subtle twitch of a finger, a faint flicker of an eyelid, or a barely perceptible movement of a toe. These small, hesitant movements are monumental victories, signaling the return of muscle control. The ventilator settings are then slowly weaned down, allowing the patient's own respiratory muscles to gradually resume their vital work until eventually the breathing tube can be safely removed. The entire process, from the moment of envenomation to full recovery, is a testament to the resilience of the human body and the unwavering dedication of medical science. It is a profound race against time, a battle waged minute by minute, hour by hour, where vigilance, rapid response, and sustained life support are the only antidotes to the silent, insidious embrace of tetrodotoxin. As the quiet understanding of this life-saving intervention gently settles within your mind, let the image of unwavering care and the slow triumph over adversity guide you deeper into a profound and peaceful rest. The antidote's elusive nature, why no cure exists. The quiet, rhythmic pulse of the ocean continues its deep, ancient song, a soothing counterpoint to the urgent drama of medical intervention we have just gently explored. We have witnessed the meticulous race against time, the unwavering dedication to life support that offers solace and survival in the face of tetrodotoxin's insidious grip. Yet, as we reflect upon this profound struggle, a crucial question often surfaces from the depths of human curiosity. If such advanced medical care can sustain life, why then is there no specific antidote, no direct cure for the venom of the blue-ringed octopus? Why does this small, potent creature continue to defy our most sophisticated pharmaceutical efforts? To understand this elusive nature of an antidote, we must first gently revisit the very essence of how tetrodotoxin, or TTX, exerts its devastating effect. As we previously observed, TTX is not a corrosive agent that destroys tissues, nor is it a chaotic disruptor of cellular machinery in a broad, indiscriminate manner. Instead, it is a molecule of astonishing precision, a finely crafted key designed to fit into one specific lock: the voltage-gated sodium channels embedded within the membranes of our nerve and muscle cells. It acts as a tenacious plug, fitting snugly into the outer opening of these vital channels, effectively jamming them shut. This blockade prevents the flow of life-sustaining sodium ions, thereby silencing the electrical signals that orchestrate every breath, every heartbeat, every thought, every movement. The body's systems are not broken, they are merely muted, held in a profound, unresponsive stillness. Now, let us consider what a conventional antidote typically achieves. Many effective antivenoms, particularly those developed for snake bites or spider bites, function by introducing specially prepared antibodies into the victim's bloodstream. These antibodies, often harvested from animals like horses or sheep that have been immunized with small, non-lethal doses of the venom, are large, complex protein molecules. Their purpose is to seek out and bind directly to the venom molecules circulating in the victim's body. When an antibody binds to a venom protein, it effectively neutralizes it, either by physically blocking its active site, preventing it from interacting with its target in the body, or by tagging it for destruction and removal by the body's immune system. These antibodies are like microscopic grappling hooks, designed to latch onto the larger, more complex protein structures of typical venoms, rendering them harmless. Herein lies the profound challenge with tetrodotoxin. Unlike the large, complex protein molecules that constitute the venoms of snakes or spiders, TTX is a relatively small, non-protein organic compound. Its molecular structure is compact, stable, and surprisingly simple in comparison to the vast, intricate architectures of protein toxins. This fundamental difference in size and composition poses a formidable barrier to the conventional development of an antidote. Firstly, because TTX is so small, it does not typically provoke a robust immune response in animals. When a large protein like a snake venom enzyme enters an animal's body, the immune system recognizes it as a foreign invader, a large, unfamiliar structure. It then mobilizes its defenses, producing antibodies specifically designed to target and neutralize that protein. However, a tiny molecule like TTX, being non-proteinaceous, often slips beneath the radar of the immune system, failing to elicit the strong antibody production necessary for creating a traditional antivenom. Without a strong immune response, there are no specific antibodies to harvest, no ready-made grappling hooks to bind to and neutralize the toxin. Secondly, even if antibodies could be generated, the way TTX interacts with the sodium channel presents another layer of difficulty. TTX binds with an astonishing affinity and specificity directly to the pore of the sodium channel. It is not merely sitting on the surface, It is deeply nested within the channel's entrance, acting as a physical molecular plug. An antidote would need to either physically dislodge this plug or somehow chemically alter the TTX molecule while it is still bound, all without causing further harm to the delicate vital function of the sodium channel itself. Designing a molecule that can safely and effectively compete with TTX for this binding site, or somehow reverse its binding without disrupting the normal essential flow of sodium ions, is an immense biochemical challenge. Any molecule that could interact with the sodium channel in such a way would carry a high risk of also interfering with the normal, healthy functioning of these channels, potentially causing severe and unpredictable side effects that might be as dangerous as the toxin itself. Consider the sheer tenacity of TTX's binding. It clings to the sodium channel with an almost irreversible grip. While the binding is not truly covalent, meaning it doesn't form a permanent chemical bond, it is incredibly strong and long-lasting. For an antidote to be effective, it would need to rapidly unbind the TTX molecules from millions of sodium channels throughout the body, or somehow render them inert while they are still bound. This is far more complex than simply binding to a circulating toxin and clearing it from the bloodstream. The toxin is already at its target, deeply embedded in the very machinery of life. Furthermore, the rapid onset of TTX poisoning, often within minutes to a few hours, means that any antidote would need to act with extreme swiftness. It would need to reach the affected nerve and muscle cells throughout the body cross-cell membranes, and then engage with the bound TTX molecules with incredible speed and efficiency. The window for intervention is narrow, and the precise, widespread binding of TTX makes this a daunting logistical and biochemical hurdle. The economic viability of developing such a highly specialized antidote also plays a role in its elusive nature. While blue-ringed octopus envenomations are severe, they are relatively rare occurrences on a global scale, particularly when compared to the hundreds of thousands of snake bites that occur annually. Pharmaceutical companies often prioritize research and development into drugs that address more widespread diseases or conditions where the potential market and return on investment are significantly larger. The immense cost, time, and regulatory hurdles involved in developing a novel drug, especially one for a rare and highly specific poisoning, can make such projects less attractive without substantial public funding or philanthropic support. Therefore, in the absence of a specific antidote, the current medical strategy remains one of unwavering, supportive care. A testament to the body's own remarkable capacity for healing, given enough time and assistance. The goal is to sustain the patient's vital functions, primarily breathing, until the body can naturally metabolize and excrete the tetrodotoxin. The liver, through its complex enzymatic pathways, slowly breaks down the toxin into less harmful compounds, and the kidneys filter these metabolites from the bloodstream, eventually eliminating them from the body through urine. This natural detoxification process is a slow, methodical journey, often taking many hours or even days, during which the mechanical ventilator acts as the patient's lifeline, patiently breathing for a paralyzed body. While there is no clinically approved antidote currently available, scientific research continues its quiet, persistent quest. Scientists explore various theoretical avenues, such as identifying compounds that might act as competitive inhibitors, meaning they could bind to the sodium channel in a way that displaces TTX, or perhaps accelerate its unbinding. Other research investigates novel enzymatic approaches, seeking to discover or engineer enzymes that could specifically break down the TTX molecule within the body. However, these approaches are still in very early experimental stages, often confined to laboratory settings, and face significant challenges in terms of efficacy, safety, and delivery to become viable therapeutic options for humans. The journey from a promising molecule in a petri dish to a safe and effective drug in a hospital setting is long, arduous, and often unsuccessful. Thus, the blue-ringed octopus stands as a powerful reminder of nature's profound chemical ingenuity, a creature whose defense mechanism continues to outwit our most advanced medical technologies. Its small size belies a chemical power that remains unparalleled in its precision and lethality, a testament to the elegant simplicity of its molecular key. The absence of an antidote underscores the critical importance of prevention of respectful distance and of immediate, unwavering life support in the rare yet profound instances of envenomation. As the quiet understanding of this elusive cure settles within your thoughts, let the intricate dance of molecular biology and human resilience guide you deeper into a tranquil, undisturbed slumber. An ecosystem's delicate balance, role, and conservation. The quiet, rhythmic pulse of the ocean continues its deep, ancient song, a soothing counterpoint to the urgent drama of medical intervention we have just gently explored. We have witnessed the meticulous race against time, the unwavering dedication to life support that offers solace and survival in the face of tetrodotoxin's insidious grip. Yet, as we reflect upon this profound struggle, a crucial question often surfaces from the depths of human curiosity. If such advanced medical care can sustain life, why then is there no specific antidote, no direct cure for the venom of the blue-ringed octopus? Why does this small, potent creature continue to defy our most sophisticated pharmaceutical efforts? To understand this elusive nature of an antidote, we must first gently revisit the very essence of how tetrodotoxin, or TTX, exerts its devastating effect. As we previously observed, TTX is not a corrosive agent that destroys tissues, nor is it a chaotic disruptor of cellular machinery in a broad, indiscriminate manner. Instead, it is a molecule of astonishing precision, a finely crafted key designed to fit into one specific lock, the voltage-gated sodium channels embedded within the membranes of our nerve and muscle cells. It acts as a tenacious plug, fitting snugly into the outer opening of these vital channels, effectively jamming them shut. This blockade prevents the flow of life-sustaining sodium ions, thereby silencing the electrical signals that orchestrate every breath, every heartbeat, every thought, every movement. The body's systems are not broken. They are merely muted, held in a profound, unresponsive stillness. Now, let us consider what a conventional antidote typically achieves. Many effective antivenoms, particularly those developed for snake bites or spider bites, function by introducing specially prepared antibodies into the victim's bloodstream. These antibodies, often harvested from animals like horses or sheep that have been immunized with small, non-lethal doses of the venom, are large, complex protein molecules. Their purpose is to seek out and bind directly to the venom molecules circulating in the victim's body. When an antibody binds to a venom protein, it effectively neutralizes it, either by physically blocking its active site, preventing it from interacting with its target in the body, or by tagging it for destruction and removal by the body's immune system. These antibodies are like microscopic grappling hooks, designed to latch onto the larger, more complex protein structures of typical venoms, rendering them harmless. Herein lies the profound challenge with tetrodotoxin. Unlike the large, complex protein molecules that constitute the venoms of snakes or spiders, TTX is a relatively small, non-protein organic compound. Its molecular structure is compact, stable, and surprisingly simple in comparison to the vast, intricate architectures of protein toxins. This fundamental difference in size and composition poses a formidable barrier to the conventional development of an antidote. Firstly, Because TTX is so small, it does not typically provoke a robust immune response in animals. When a large protein like a snake venom enzyme enters an animal's body, the immune system recognizes it as a foreign invader, a large, unfamiliar structure. It then mobilizes its defenses, producing antibodies specifically designed to target and neutralize that protein. However, a tiny molecule like TTX, being non-proteinaceous, often slips beneath the radar of the immune system, failing to elicit the strong antibody production necessary for creating a traditional antivenom. Without a strong immune response, there are no specific antibodies to harvest, no ready-made grappling hooks to bind to and neutralize the toxin. Secondly, even if antibodies could be generated, the way TTX interacts with the sodium channel presents another layer of difficulty. TTX binds with an astonishing affinity and specificity directly to the pore source of the sodium channel. It is not merely sitting on the surface, it is deeply nested within the channel's entrance, acting as a physical molecular plug. An antidote would need to either physically dislodge this plug or somehow chemically alter the TTX molecule while it is still bound, all without causing further harm to the delicate, vital function of the sodium channel itself. Designing a molecule that can safely and effectively compete with TTX for this binding site, or somehow reverse its binding without disrupting the normal essential flow of sodium ions, is an immense biochemical challenge. Any molecule that could interact with the sodium channel in such a way would carry a high risk of also interfering with the normal, healthy functioning of these channels, potentially causing severe and unpredictable side effects that might be as dangerous as the toxin itself. Consider the sheer tenacity of TTX's binding. It clings to the sodium channel with an almost irreversible grip. While the binding is not truly covalent, meaning it doesn't form a permanent chemical bond, it is incredibly strong and long-lasting. For an antidote to be effective, it would need to rapidly unbind the TTX molecules from millions of sodium channels throughout the body, or somehow render them inert while they are still bound. This is far more complex than simply binding to a circulating toxin and clearing it from the bloodstream. The toxin is already at its target, deeply embedded in the very machinery of life. Furthermore, the rapid onset of TTX poisoning, often within minutes to a few hours, means that any antidote would need to act with extreme swiftness. It would need to reach the affected nerve and muscle cells throughout the body cross-cell membranes, and then engage with the bound TTX molecules with incredible speed and efficiency. The window for intervention is narrow, and the precise widespread binding of TTX makes this a daunting logistical and biochemical hurdle. The economic viability of developing such a highly specialized antidote also plays a role in its elusive nature. While blue-ringed octopus envenomations are severe, they are relatively rare occurrences on a global scale, particularly when compared to the hundreds of thousands of snake bites that occur annually. Pharmaceutical companies often prioritize research and development into drugs that address more widespread diseases or conditions where the potential market and return on investment are significantly larger. The immense cost time and regulatory hurdles involved in developing a novel drug, especially one for a rare and highly specific poisoning, can make such projects less attractive without substantial public funding or philanthropic support. Therefore, in the absence of a specific antidote, the current medical strategy remains one of unwavering, supportive care. A testament to the body's own remarkable capacity for healing given enough time and assistance. The goal is to sustain the patient's vital functions, primarily breathing, until the body can naturally metabolize and excrete the tetrodotoxin. The liver, through its complex enzymatic pathways, slowly breaks down the toxin into less harmful compounds, and the kidneys filter these metabolites from the bloodstream, eventually eliminating them from the body through urine. This natural detoxification process is a slow, methodical journey, often taking many hours or even days, during which the mechanical ventilator acts as the patient's lifeline, patiently breathing for a paralyzed body. While there is no clinically approved antidote currently available, scientific research continues its quiet, persistent quest. Scientists explore various theoretical avenues, such as identifying compounds that might act as competitive inhibitors, meaning they could bind to the sodium channel in a way that displaces TTX, or perhaps accelerate its unbinding. Other research investigates novel enzymatic approaches, seeking to discover or engineer enzymes that could specifically break down the TTX molecule within the body. However, these approaches are still in very early experimental stages, often confined to laboratory settings, and face significant challenges in terms of efficacy, safety, and delivery to become viable therapeutic options for humans. The journey from a promising molecule in a petri dish to a safe and effective drug in a hospital setting is long, arduous, and often unsuccessful. Thus, the blue-ringed octopus stands as a powerful reminder of nature's profound chemical ingenuity, a creature whose defense mechanism continues to outwit our most advanced medical technologies. Its small size belies a chemical power that remains unparalleled in its precision and lethality, a testament to the elegant simplicity of its molecular key. The absence of an antidote underscores the critical importance of prevention, of respectful distance, and of immediate, unwavering life support in the rare yet profound instances of envenomation. As the quiet understanding of this elusive cure settles within your thoughts, let the intricate dance of molecular biology and human resilience guide you deeper into a tranquil, undisturbed slumber. Whispers from the deep, reflections on beauty and lethality, the quiet, ceaseless rhythm of the ocean, a profound and ancient breath, now gently draws our journey to a close. Yet the echoes of our exploration linger soft and persistent within the tranquil chambers of the mind. We have navigated the intricate depths of the blue-ringed octopus's world, from its initial camouflaged whisper within the reef to the electrifying declaration of its azure warning, and through the silent molecular dance of its potent venom. As the last scientific facts softly settle, we are left with a deeper contemplation, a quiet reflection on the profound duality embodied by this small, luminous creature, its breathtaking beauty interwoven with an undeniable inherent lethality. Imagine once more the sun-dappled shallows where coral branches reach like ancient gnarled fingers towards the light. Here, our small octopus, often no larger than a child's fist, moves with an almost ethereal grace. Its 8 arms, fluid and sensitive, propel its mantle with a liquid elegance, allowing it to glide through the water or meticulously crawl across textured rock it suckers, testing every surface, gathering silent intelligence from its surroundings. When undisturbed, its skin, a masterpiece of natural camouflage, ripples with muted tones of earthy brown, soft grey and sandy yellow, allowing it to vanish almost entirely against the variegated backdrop of the seabed. It is a living phantom, a master of blending, a testament to the quiet artistry of survival. Its large, dark eyes, perpetually open to the liquid world, hold a silent intelligence reflecting the ancient wisdom of its cephalopod lineage, a gaze that seems to absorb the very essence of the ocean's mysteries. In this tranquil state, it is a creature of exquisite, understated beauty, a jewel crafted by the patient hand of evolution. Yet this gentle beauty harbors a profound, silent power. The sudden incandescent flash of its electric blue rings, a vivid pulsating spectacle against a rapidly darkened body, serves as nature's most unambiguous warning. This transformation from quiet observer to a beacon of danger is a chilling reminder that true potency often resides in the most unexpected forms. The tetrodotoxin, an invisible weapon crafted by its symbiotic bacterial partners, is a testament to the ocean's capacity for complex, sophisticated lethality. It is not a malevolent force, but a tool honed over millennia, an essential mechanism for both predation and defense, ensuring the survival of a soft-bodied creature in a world teeming with both larger predators and agile prey. This paradox, the delicate beauty cradling such an absolute silent power, invites a profound respect, a recognition of the intricate balance woven into the fabric of life itself. Within the vast, interconnected network of its Indo-Pacific home, the blue-ringed octopus plays a vital, albeit often unseen role in the delicate balance of the reef ecosystem. As a proficient predator primarily preying on small crabs, shrimp, and tiny fish, it helps to regulate the populations of these creatures. Its presence therefore contributes to the overall health and stability of the reef, preventing any single species from becoming overly dominant and ensuring a diversity of life forms. Without such specialized predators, the intricate food web could unravel, leading to unforeseen consequences throughout the delicate marine environment. It is a small but significant cog in the grand machinery of ocean life. Each bite, each hunt, a subtle contribution to the ebb and flow of energy through the ecosystem. Conversely, despite its formidable venom, the blue-ringed octopus is not entirely immune from the cycle of life and death. Scientists believe that certain larger marine predators, such as some species of moray eels or larger fish, may possess a degree of immunity or tolerance to its venom, or perhaps employ hunting strategies that minimize their risk of envenomation. This means that even this highly venomous creature is part of a larger food chain, its existence intertwined with the lives of others, both those it consumes and those that might on rare occasions consume it. This intricate dance of predator and prey, of defense and counter-defense, speaks to the dynamic, ever-evolving nature of marine ecosystems. However, the delicate balance of the blue-ringed octopus's world, and indeed the entire marine ecosystem, faces unprecedented challenges from human activities. The very coral reefs that provide its essential shelter and hunting grounds are under immense pressure. Rising ocean temperatures, primarily driven by climate change, lead to coral bleaching, a devastating phenomenon, where corals expel the symbiotic algae that provide them with sustenance, often leading to their demise. Ocean acidification caused by the absorption of excess carbon dioxide weakens the very structure of coral skeletons, making them more vulnerable to erosion and damage. Pollution from plastic debris that chokes marine life to chemical runoff that poisons the water further degrades these vital habitats. Overfishing, both targeted and incidental, can deplete the populations of crabs, shrimp, and small fish that form the octopus's essential diet, leading to food scarcity and increased stress on the species. The conservation of the blue-ringed octopus, therefore, is inextricably linked to the broader health of its reef and coastal environments. Protecting these habitats means addressing the root causes of climate change, reducing pollution, and implementing sustainable fishing practices. It means fostering a deeper understanding and respect for all marine life, recognizing that even the smallest, most potent creatures play a crucial role in maintaining the health and biodiversity of our oceans. Education is paramount, ensuring that coastal communities, divers, and tourists are aware of the octopus's presence, its beauty, and its danger, promoting respectful observation rather than intrusive interaction. The goal is not to eradicate or fear, but to coexist, acknowledging the inherent value of every species, every living thread in the vast, interconnected web of life. The blue-ringed octopus, with its hidden life and its flashing azure warning, serves as a poignant emblem of the ocean's enduring mysteries and its profound vulnerability. It is a living testament to the wonders that still lie beneath the waves, a creature that continues to challenge our understanding and inspire our awe. Its story, from its silent discovery to the molecular secrets of its venom, is a whisper from the deep, reminding us that the natural world holds countless marvels yet to be fully comprehended, countless delicate balances that deserve our unwavering protection. As these reflections on beauty and lethality, on ecological roles and conservation, gently settle within your mind, allow the quiet contemplation of the ocean's vast, dreaming depths to guide you into a profound and peaceful stillness. And so, as we drift away from the shimmering, mysterious depths where the blue-ringed octopus holds its silent power, let the echoes of its hidden beauty and potent secrets gently dissolve into the quietude of your own inner ocean. Just as this small creature retreats into its coral sanctuary, blending seamlessly with its world, allow your thoughts to recede, finding their own peaceful refuge within the soft, yielding contours of your consciousness. The ocean's whispers, carrying tales of life's intricate balances and the profound quietude of the deep, now become the gentle currents that lull your mind. Imagine your awareness like a soft, luminescent creature sinking slowly through layers of serene water, leaving behind the day's light and sound. Perhaps in the expansive, silent dreamscape, you will encounter your own silent jewels, hidden wonders waiting to unfurl in the tranquil darkness, bathed in a soft, internal glow. May the deep, undulating currents of the vast, dreaming ocean cradle your spirit, guiding you to a slumber as profound and undisturbed as the ancient silent sea itself. Sleep well, goodbye, and see you again.